Thank you, Dawn, so much. And thank you, Madeline, for allowing me to be a part of this conference. Thank you all for being here. This is, for me, very humbling because there's so many people in the room who have taught me over the years and inspired me. And you'll be hearing from them all soon. So when I was a neonatology fellow, I read this paper by Nigel and John Kiley about follow-up methods, and I thought there's another neonatologist interested in the same topics I'm interested in. And then he launched the New Jersey brain hemorrhage study, or neonatal brain hemorrhage study, and I think Jennifer's on the program today to speak. And I was further inspired, and then he wrote my favorite book, and my favorite chapter is Prognosis, and I've read the Prognosis chapter 10 times or more. So my particular part of the universe that Nigel has contributed so much to is the extremely low gestational age newborn study. So I'm going to spend about 5 minutes telling you about that study. As this audience knows, individuals born extremely preterm, although constituting only 1% of births in the US or less, account for a disproportionate number of children with developmental disabilities. And equally important, their survival has increased almost 20-folds in recent decades. And around 1999, Alan Levinson, another mutual friend, began launching this study. And this was an early investigators meeting. You'll see Nigel in the front, and Purdue, who's on the call, is sort of in the middle. Alan Levinson, you'll hear from later. And we all look about the same. And we launched this study in 2002 in 5 US states, which I think was an accomplishment in and of itself, and really is attributable to Alan and Nigel's vision to have a large enough network to recruit 1,500 neonates. And it was one of the earlier, I think, biomarker studies in neonatology. So another innovation, we included a number of biomarkers of inflammation, and that was a central biologic mechanism on which we were focused. And we used neutrophils and placenta proteins in neonatal blood, including not just inflammatory proteins, but proteins that we envision as perhaps being protective. And since then, we've added a number of omics, thanks again to another innovation. That was the foresight that Nigel and Alan had to archive biospecimens. And so we've now, Nigel may not even know this, we've retrieved the umbilical cords from the freezers and measured metals in those. And the maternal blood spots have gone to measure organic chemicals in the mother's blood. So a lot of vision there through Nigel and Alan's wisdom and intellect. In the first phase, we focused on brain ultrasound. This would not have had the excellence that it has were it not for Nigel's earlier New Jersey brain hemorrhage study, which really, the methods of which guided what ELGAN did. We saw the children at 2, again at 10. Recently, we've started to publish on the 15-year outcomes, and we're almost finished with the 17-year outcomes. And I'm just going to show one result slide. I think this is the only one. And this is sort of the main theme of this first phase of the ELGAN study, where if you move from front to back, you're moving from less inflammation to more inflammation, and you see the rate of cognitive impairment increase. And then if you move from left to right, you see this is an increased number of neurotrophic proteins, and you see the risk of cognitive impairment decline. So some support for this overarching hypothesis that inflammation may be an important contributor to brain-related disorders. We also in the data examined this multi-hit hypothesis that I think this audience is very familiar with, and we used as a prenatal marker, we use both microorganisms in placenta parenchyma as well as neutrophils in placenta. And in this very nice study by Diana Yonke, we found on the far right that children who had both a prenatal and a postnatal biomarker of inflammation had the highest risk of ventricular enlargement. So in the last phase of, or in the most recent phase of the ELGAN study, we've partnered with ECHO, and I think you are all familiar with this. Gene Curver's a partner in this ECHO project. And we're charged with answering crucial questions about the effects of a broad range of early environmental exposures on child health and development. This is a complicated conceptual model, but essentially on the left, you'll see a variety of prenatal exposures. On the right, a range of outcomes. And in the middle, these biologic response mechanisms of inflammation and epigenetics that we're focused on. And then in both of these eras connecting the left and the right, we have sex differences as a potential effect measure modifier. And we've begun to get some data from our placental epigenetic work. This is, I think, one of the potentially more exciting findings where a graduate student in Rebecca Fry's lab, my collaborator at UNC, looked at the relationship between CpG DNA methylation and placenta in relation to two outcomes, one being inflammation on the first day of life, which intuitively you would think might be the stronger relationship, and secondly, to inflammation present more or less through the first 2 weeks of life. And long story short, on the left is the plot for the day one inflammation, on the right for sustained inflammation, and much stronger association with sustained inflammation. And the reason that's relevant is that right in the center of the slide, you'll see sustained inflammation. And we found repeatedly that that's the more strongly associated type of inflammation with later on neurodevelopmental outcomes. So this at least provides some proof of concept that this DNA methylation in placenta might be contributing through sustained inflammation in the neonate's blood to later impairments. Lastly, I'll just show you a potential prenatal exposure that might be operating through this type of a mechanism, and that's microorganisms in the human placenta. This is the heat map for the CpG methylation on the left for lactobacillus in the placenta, and on the right for strep. Very different, so very different CpG signatures. And if you then look at the genes that are encoded by the differentially methylated genes, you see it enriched in inflammatory pathways like the NF-kappa B pathway. In work that Raina Ficharova led, we found also that a similar picture was true if you look at the relationship between lactobacillus and placenta and inflammation in the baby's blood. And then most recently, lactobacillus in placenta is associated with a decreased risk of cognitive impairment. So again, sort of a proof of concept that there may be events occurring and exposures operating in the placenta that have very long-term consequences. Yeah, so I'll just end by thanking Alan Leviton, who I hope is on the call. He led the ELGAN-1 study. He's been an inspiration to Nigel, an inspiration to me, and to many people in this room. Carl Kuban, our colleague in Boston, who led the second phase. And then as you can see in the left lower corner, the group's grown quite a bit over the years. And I think this is in much part due to the priority that Nigel and Alan have always placed on fostering the next generation of investigators. So thank you so much for allowing me to be a part of this symposium, and I'll acknowledge the funding sources.